everyone, and welcome to the journal webinar series of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. This series features the latest research published in our society journals. I'm Liz Gebhardt with Crop Science. Thank you for joining us today for protecting plant germplasm from climate change. Before we begin, I'd like to mention that questions can be entered in the Q&A section, and the moderator will address them at the end of the presentation. The webinar will be recorded and the recording will be emailed to all attendees after the session. Now I'm happy to introduce our moderator for today's webinar, Dr. Paul Scott, the editor of Crop Science. Dr. Scott has been a research geneticist in the USDA ARS in Ames, Iowa for 27 years. His research focuses on corn improvement, including basic and applied studies of corn genetics and breeding. His current projects involve understanding genetic systems that control cross-pollination of corn and increasing the levels of nutritionally limiting amino acids in grain. Paul?
Well, thank you, Liz, and thank you to everybody who's joining us virtually today for taking the time to join us today. As Liz mentioned, please enter any questions you have in the chat, and we will address those at the end of the presentation. Without further ado, I'd like to introduce today's speaker, Dr. Brian Irish. Brian was trained as a horticulturist and plant pathologist and has spent his professional career with the USDA ARS National Plant Germplasm System. 11 of those years were with the Tropical Agriculture Research Station in Mayagüez, Puerto Rico, where he led a team that managed agriculturally important clonally propagated tropical plant genetic resources. For the past 8 years, he has been with the Plant Germplasm Introduction and Testing Research Unit, leading efforts to conserve and promote the use of over 1,300 temperature-adapted forage legume accessions. Today, Dr. Irish will speak about the dangers of climate change to plant genetic resources management in the United States and potential methods that may be used to protect agriculturally significant plant germplasm. This talk is based on a report that was published in the July 2023 issue of Crop Science. Dr. Irish, welcome, and the floor is yours.
Thank you. Can you hear me? And thank you for that introduction, Dr. Scott. Could you please let me know that you can hear me?
Yes, we can hear you.
Okay, great. And would you let me know that you can see my slide?
Yes.
Okay, great. So again, thank you for the introduction, and for those of you joining us virtually. I wanted to take a moment to acknowledge the Crop Science Society of America for this opportunity, for those of us who were involved in this essay, to discuss how plant genetic resources are being impacted by climate change. I also wanted to acknowledge some of our co-authors on the essay, in particular, Drs. Gail Volk and Peter Bredding, who spearheaded the effort and have provided the template that I've used and I've adapted for today's slide deck. The impetus for the essay came from an invitation by editor Erwin Goldman on behalf of the Tri-Societies on a special issue on climate change. And it's very appropriate, very timely to be discussing climate change. It's affecting us all, and it seems to be doing so more frequently, more intensely, and it's affecting how we manage plant genetic resources. So on the title slide, that figure off to the right is something that I've adapted from, or I've taken from the USDA Climate Solutions landing page. And it outlines sort of the process that we used to develop the essay, and it outlines the presentation as well, where we are aware of climate change. We're beginning to assess where climate change impacts are being felt the most in the National Plant Germplasm System. We're planning and we're beginning to discuss implementation strategies, and later we will focus on monitoring and evaluating to see if those implementation strategies are mitigating the effects of climate change. So more specifically, the essay and this presentation focuses on describing the potential effects of climate change on the USDA ARS National Plant Germplasm System, National Plant Germplasm Systems, Plant Genetic Resources. And so I'll use that NPGS acronym and the PGR acronym throughout the presentation. And another term that's synonymous with plant genetic resources is germplasm. And so in order to develop this assay and begin to assess the impacts, we developed an application providing US temperature and precipitation predictions for these NPGS gene banks. And in the essay, as well as in the presentation, we'll provide some ex situ and in situ examples or case studies demonstrating instances of climate change impacting the current collections as well as potential impacts in the future. And we wrap up by describing possible NPGS management responses to climate change and its effect on the germplasm collections, infrastructure, and our overall operations. It's likely that many in the audience are familiar with the National Plant Germplasm System, but I thought it was appropriate to include at least one slide giving a brief overview of this organization that consists of about 20 different sites. Most of these are gene banks managing collections across continental United States and insular regions, including Hilo, Hawaii, and Maya, West Puerto Rico. And they're superimposed here on this plant zone hardiness map. And the reason Pullman location is highlighted is because that's the site that I'm affiliated with. But so the NPGS conserves over 620,000 unique entries or accessions in the collections of over 16,000 unique species. And these are collections of agriculturally important crops, but also many, many of their wild relatives. And so the NPGS and its gene banks have this long-standing partnership with land-grant universities and state agricultural experiment stations, as well as with commodity groups and agricultural industries in those regions where those crops assigned to those gene banks grow well and do well. And I wanted to make a point about the NPGS and the genetic resources, and that is that those of us that are involved in managing them, but I think pretty much everyone understands that this is one of the very important tools that agriculture can leverage in order to continue adapting to the ever increasing number of stressors, the abiotic and biotic. And this is done by identifying the genes and the traits, and then by plant breeding, incorporating those into modern improved agricultural cultivars and varieties. So I've already mentioned that we developed an application in order to help us assess the impacts of climate change. And the application is called the NPGS Climate Futures application and can be accessed by that link down in the bottom. And we, again, relied heavily on one of our co-authors, Daniel Carver, with his expertise on managing large data sets and climate data and geographic information systems to help us develop this browser-based tool that tries to predict the temperature and precipitation of 31 different locations or 31 different ex situ gene bank locations and 6 in situ sites. And I might be confusing you with the number of gene bank locations that we saw on the map a moment ago, which was about 20, and there's 31 different sites that the Climate Future applications is using for ex situ gene bank locations. And that's because some of the gene banks might have multiple research farms. And so we also included the 6 in situ sites that are affiliated to NPGS. And so the Climate Futures application uses historical data for the gene bank locations from 1970 to 2000, as well as these greenhouse gas emission prediction scenarios based on models and policies, national, international, that are developed by the Intergovernmental Panel on Climate Change, IPCC. And we don't have to get into any more details here. There'll be some examples in the following slides of the outputs. But one of the neat things about this application is that anyone that has access to the IPCC climate scenarios and has a georeference location could potentially leverage or use this tool to estimate what the climate might be at their specific location. So it's a tool that could be used by the community. So by now, it should be obvious that the task of managing large germplasm collections like those of the NPGS takes a lot of effort. It's a very technical process, and it's because of the large number of accessions and the diversity in the collections. Well, if you add in some unpredictable climate and weather events, it makes that job much more difficult. And so the next slides are examples, again, of how climate change is impacting us on the ground, in the locations, in the gene banks. The image on the left-hand side of your screen are of insect-proof isolation cages at the Prosser, Washington location where I'm located, where we manage temperate adapted forage legumes, crops like alfalfa and clover and trefoil. But the insect-proof cages are there for us to introduce, manage insect pollinators to help us seed set and regenerate germplasm that's queued for regeneration because the viability is declined or because we've been distributing a lot of it. And also those cages help prevent those pollinators from moving between cages, preventing genetic erosion and maintaining the genetic integrity of those unique accessions. So contrast that to the picture on the right-hand side, which is a similar situation where sunflowers are being, Helianthus species are being regenerated in the Ames, Iowa site. And that's after a nasty climatic event called the derecho, with, I think it was about 100-mile-an-hour straight winds, and those cages and the regeneration stood no chance, and it was a loss for them. And so the Iowa State University News Service, the headline there was a derecho debrief. Not even a scientist storm chaser expected this August 10th storm. And so climate events, weather events like this ones are predicted to increase, affecting our operations. So another example from that same site in Ames, Iowa, they hold and manage the large maize germplasm collection. And the graph or figure on the left-hand side of your screen was prepared by maize curator Vivian Bernal. And if you observe or look at the y-axis, those are harvested years or attempted pollinations, and then the x-axis is the daily maximum temperature increasing as you go to the right. And very clearly, at about 34 degrees centigrade, the pollination success drops precipitously. And unfortunately, this is the output from the NPGS Climate Futures application on the right-hand side, with that historical temperature and precipitation, or in this case, temperature data in the dark blue for the 30-year period, and then the predictions in 20-year intervals for those 4 climatic scenarios. And I did forget to mention those 4 climatic scenarios go from sort of the best case scenario where greenhouse gas emissions are mitigated, more sustainable approaches, to the worst case scenario in the red bar, where greenhouse gas emissions are predicted to rise based on use of fossil fuels. And so if you look at that dotted line, that's about that 34-degree threshold. It looks like in the future, the Ames, Iowa site will experience warmer temperatures that will likely affect their corn or maize regenerations. So on some of these slides, I've added a little asterisk and a comment at the bottom, just to indicate that we've thought about how to mitigate, how to deal with these scenarios. And in this case, there's potential to alter planting dates of these maize accessions or germplasm that might be more susceptible to that summer heat. But when one alters planting dates, then that brings on another set of problems often. In a similar situation, but then in a different geographic area in the United States, in the early summer of 2021, we've experienced in the Pacific Northwest a very difficult heat dome with 46 degrees centigrade in eastern Washington early in the season, causing numerous pulse regenerations to fail. And so pulse crops are chickpeas, lentils, fabas, and peas. And the image there is of our Central Ferry Research Farm, where a lot of the regenerations occur. So again, the output from the NPGS Climate Future applications shows that if we don't do something to mitigate the greenhouse gas emissions, it's likely temperature is going to rise more and affect some of the pulse regenerations. And this is despite irrigation, and I highlighted that word, or underlined it, because I wanted to make a point. At both the Pullman Research Farm as well as the Central Ferry Farm, we've updated and installed irrigation. But in this case, it was ambient air temperatures, and irrigation wouldn't have helped us much. So similar to the situation with maize, it's possible that for the more sensitive materials to this heat, these increasing temperatures, it's possible that we might be able to regenerate or plant them at a different time, or also possibly regenerate some of that germplasm in screenhouses and greenhouses. Some of the examples that we've seen so far have been for crops that are propagated by seed. Unfortunately, the clonal germplasm collections are being impacted even more so. And so in this example, we see an image on there on the screen on the left of prunus or stone fruit collections at the Davis repository in California, where they're experiencing incomplete dormancy, delayed pollination, and foliation with reduced yield and performance. And in the output from the NPGS Climate Futures application to the right, this is a different bioclimatic factor output. This is the mean temperature of the coldest quarter. And what's happening is that the temperature during the winter is not as cold as it once was, and the temperatures are predicted to increase, and it will affect plant physiology and hinder backup efforts, especially for prunus. And so I like what the research leader for that site, Claire Heinert, said. So that impacts on these collections is sort of a hidden impact, and it's because the dormant buds are not reaching ideal dormancy levels, and then the germplasm cannot be backed up effectively. And so one of the strategies for this collection in particular is to decide to possibly duplicate part of the prunus collection in Geneva, New York, to improve health and supply that deeper dormancy that is needed for those buds to be cryopreserved. It's ironic that I transitioned from the slide in Davis, California to this one on Geneva, New York, where I said the plans are possibly to move some of that susceptible germplasm and the stone fruit collection to Geneva, New York, where they themselves are facing some problems brought on or intensified based on warming temperatures and wetter spring environments. And so the apple collections in Geneva, New York have been devastated by this streptomycin-resistant Erwinia amylavera, the causal agent of fire blight. And the predicted future in this location are wetter springs with warmer springs, and this may lead to bigger losses. And so when I spoke to Ben Gutierrez, who is the manager of these collections, he pointed out there's sort of a silver lining in this situation. And that is, as diseases, for example, fire blight in this collection, that are intensified because of the climate change, that there's a potential then to be able to phenotype the collection. And in this case, they were able to phenotype the germplasm collection and publish, and that's a agronomy journal publication page showing, contrasting a very susceptible apple accession to the one on the right. And they've identified potential sources of disease resistance to this disease in the collection that could potentially be used in plant breeding. And so the management in this case might include heavy pruning, which is what you see on the picture on the left. But also, one of the really important things is continue to ensure that all of the malus species or apple germplasm are backed up effectively. And the examples so far have been of temperate region collections of the world, and that's where climate change is affecting mostly, but tropical and subtropical regions of the world are also not immune, and the collections that are maintained there are also being affected. And so the National Plant Germplasm System is in the process of establishing a new coffee collection that will be maintained at the Gila, Hawaii, and Maya West Puerto Rico sites. And both of these islands, coffee leaf rust has been reported and has been spreading, especially due to these warming trends. And so the gene bank managers there will have to use site selection and optimize our management in view of these newly reported diseases. So they, in order to manage the collections. The examples so far have been of specific locations or specific crops, but there are some other general impacts to NPGS management of collections. And so we, in developing the assay, we looked into the literature, and it was very clear that some insect and arthropod pests are favored by these warmer temperatures. And that's because the temperatures in the winter won't be as severe, and so the populations would overwinter and cause earlier infestations and expanded multiplication rates and generation numbers. And we found in the literature this is especially true for some of the lepidopteran pests that are very important agricultural pests and also of the plant genetic resources. So with increased pest pressure often comes many important vector diseases. And just generally, if the plants are stressed already because of maybe higher temperatures, these are also predisposed to insect and arthropod pests and diseases. And so we'll have to, as managers, we'll have to be aware and detect these earlier and implement integrated pest management strategies to be able to manage the collections more effectively. So not all insects are detrimental. We already mentioned that we use insects as managers to help us with the seed set. And so the pollinators, in this case, honeybees, bumblebees, and we use alfalfa leaf cutters, are often less active at low temperature, but when the temperatures really are high, they dedicate a lot of their resources to cooling the brood or the hives and not towards pollinations. And so these insects, as they're stressed, also causes declines in their populations. Another point to make about the phenology of plants, the physiology and phenology of plants, oftentimes stressed plants might flower earlier and there might not be that synchrony between the pollinators and the flowers. And then oftentimes in integrated pest management, we use biological control organisms, and these might be affected by changing climate with lower reproductive rates at higher precipitation, where precipitation is already high, or where temperatures are expected to increase significantly. This is the only slide that addresses in situ conservation, and I mentioned earlier that the NPGS is involved with 6 different sites in the US of native crops or crop wild relatives. And in this example, there's a proposed native in situ cranberry or vaccinium reserve site. And this area will need to be monitored in case the population shift outside of the boundaries that have already been designated for this native in situ site. I also just touched on momentarily about the idea of backing up germplasm, which is one of our core missions. And so for one of the really good things about backing up germplasm is that we have a location in Fort Collins, Colorado called the National Laboratory for Genetic Resources Preservation, and they effectively back up, I think it's like 82% of the seed propagated collections. And so that's really good, but for the clonal collections, it's a little bit more difficult. And so again, we strive to back up most of the germplasm of this site in Fort Collins, but also a secondary site. Most of the seed germplasm is also going to be backed up at Svalbard Global Seed Vault in Norway. For clonal collections, they could be replicated as safety duplicates at different locations and different farms, but also they could be in vitro propagated and stored at lower temperatures. And in some cases, meristematic tissues, even for clonal material, can be used to back up germplasm. And that's the case for 2 really important clonal germplasm collections. That's the Malus collection, which is backed up and cryopreserved, as well as the citrus collection. And the folks at NLGRP continue to research on effective ways of backing up germplasm, both in the seed form and clonally. In an effort that's spearheaded by Gail Volk and Patrick Byrne of Colorado State University and others, there is a training resource that is being developed on a platform called Grin University. And this platform has a lot of training materials that are geared towards trying to help with that gap that's being that's coming on, with a lot of folks retiring from the NPGS and new staff coming on board, needing to learn a lot about how to manage plant genetic resources collections. And within the materials there's a lot of the materials that are focusing on how climate change is affecting the management of the collections, including ebooks and ebook chapters that can be accessed freely from the GreenU site. And so climate change is something that everybody, many different organizations at all different levels are beginning to address and discuss and plan for. And so here are just some examples of some of the materials that our agency, the Agricultural Research Service on the left, and then our department have developed to address some of the effects of climate change. And this is not just at the US level, but internationally. Again, the Intergovernmental Panel on Climate Change has come up with guidelines and processes for dealing with climate change. And these are all resources that we in the NPGS can leverage to develop plans for mitigating the effects of climate change. So I'm going to wrap up with some of the sort of recommendations or the plans for the NPGS. And the idea is that the NPGS would like to continue its service mission of making plant genetic resources accessible to the stakeholders and to provide these genes and traits, especially for breeding modern cultivars that can adapt to climate change. We plan on monitoring the plant genetic resources for environmental change response while trying to take advantage of identifying the traits that are valuable in the collections. And so as resources allow it, we would also like to continue evaluating plant performance under this wide range of conditions across the NPGS. We also want to assess if current NPGS gene banks meet the current and future needs in order to decide if collections or portions of the collection need to be moved, for example. We want to redouble or focus especially on backing up or duplicating clonal germplasm collections, and that's especially true for the sensitive materials in the clonal gene banks. In addition, we plan to add irrigation capacity where gene banks would benefit. I didn't touch on this next point during the presentation, but in the essay we talk about how important it is to use alternative power sources to gaining cooling and heating efficiencies. Anything we can do to mitigate possible greenhouse gases in the future would help us in managing the germplasm collections and help everyone. So we also do plan on monitoring those native in situ plant genetic resource populations and the in situ germplasm resource reserves. And we also want to make sure that we use or that each site use or is affiliated with meteorological stations to inform us about how climate change might be affecting us to input into the Climate Futures application. And we continue to make an effort to develop plans to respond to climate change scenarios as they arise. And so with that, I'm wrapping up my short presentation. I've listed some information here, and maybe if there is a take-home message that I want the audience to have is that it is complicated to manage the large germplasm collections, but we're going to make all the efforts we can to address keeping these collections long-term and making them available. And I'm optimistic that we'll meet the challenge of being able to face these climate change impacts. Thank you very much.
Thank you, Brian. That was a fantastic presentation. I just want to remind all the listeners that if you have any questions, you're welcome to type them into the chat, and we'll try to get to them all as they come in. I just want to start, Brian, by asking, you made it very clear that the climate change is going to have a big impact on your job and the way you do business. I wonder if you could say a little bit about how these impacts of climate change are going to affect your customers, those of us who depend on the National Plant Germplasm System resources. And do you have any guidance for us on how we can help you with this, with the new problems that are arising.
Yes, I'm mulling your question over, make sure that I understand, but you want to know how stakeholders might be affected by us being affected, and so, and how you might be able to help. So sure, spreading the information, creating awareness of how this is impacting everyone, including the national plant germplasm system. It would be very important. That's one way to help. So I have a feeling that as climate change impacts our activities, we might have to restrict availability of some of the germplasm until we may be able to come up with contingencies for managing them. I mean, just putting in the context, let's say someone is requesting a forage legume that we manage, and we oftentimes only distribute small amounts of seed. We might have to reduce the amount of seed because we're not being so effective at regenerating them. The clonal germplasm collections are, requested, and it's scion wood or propagated material, and so that might not be available at that time. There might have to be some patience taken on by the stakeholders. Yeah, I remember having invited one of our co-authors, Marilyn Warburton, to our webinar today, and if she was on, or if any of the other co-authors are on and maybe could respond also in the chat in case I messed something up during the presentation, or that they have some input on some of your questions.
Well, that was a good answer. Thanks, Brian.
Sure.
You gave some good examples of events that have happened. We're already seeing impacts of climate change at different locations, and it seemed to me like they were scattered pretty much throughout the country. Are there specific locations that you anticipate will be affected more than others, or can you predict even what locations might be most impacted by the effects of climate change?
That second part of the question is the trickiest one. Can one predict? Yes, one can try to predict, but things obviously change. And I think in the essay and in the presentation, I alluded to the fact that it's possible, or is likely, that the more temperate located gene banks are the ones that are going to be mostly impacted, and that's because it's the polar regions of the world that are seeing the effects of global warming or climate change more severely. And so I don't think that the subtropical and more southern location latitude gene banks are not going to be impacted, but it's likely the clonal germplasm collections in particular are ones that we really have to keep very, very careful and monitor them. And I think they might be the ones that be most impacted in temperate regions.
Yeah, so I guess similar question. You mentioned the risk to clonal collections. Are there specific crops then that you're particularly worried about? What are the crops that are at most risk?
Mm-hmm. I also touched on that both the apple and the citrus, even though it wasn't an example in the presentation, citrus collections are threatened because of fire blight and climate change, and just the plants are unhappy, but also citrus greening in particular for the citrus germplasm collections. And both of those, the folks at Fort Collins, Colorado, have come up with very ingenious ways to use meristematic tissues to cryopreserve those collections. And I believe, for the most part, they're both backed up in Fort Collins. And so, yeah, some of the clonal germplasm collections are especially threatened. Because I'm affiliated, or was affiliated, with the tropical site in Mayagüez, Puerto Rico, banana germplasm is extremely important to the world, to that region, culturally, economically. And there's some nasty diseases in the soil, fusarium wilt in particular, that could affect that germplasm. And so it's important to have that also backed up. It's a crop that can be backed up in tissue culture in the medium term.
So you mentioned briefly a collaboration with the Germplasm Center in Norway. Are there other international collaborations that you're going to try to expand, or in other organizations you'll engage with to try to mitigate the effects of climate change?
I wish I had a good answer. I'm also thinking about this one. I am certain that all gene banks are facing similar challenges like we are. Large numbers, diverse crops, and add climate change. And again, those gene banks in locations in northern or southern latitudes, not necessarily in the equatorial regions of the world. Yeah, they'll have similar problems. Off the top of my head, I can't think of that we're working directly with any one organization, but we would love to, bring our heads together to address this, especially if there's a lot of commonalities in these challenges. I would assume, gene banks, part of the CGIAR system, and some of the national gene banks in Europe would be great collaborators.
Good to know. Okay, well, I am not seeing any more questions in the chat, and I just want to check with the organizers to make sure that I'm not missing anything. If someone could confirm that we've addressed all the questions in the chat, that would be helpful.
Few, let me send them. Shay and I will send them over real quick.
Yeah.
Thanks, Liz. We did have a question about continuing education credits in the chat, and that was addressed in the chat.
Perfect.
If you're interested, you can see that.
Yeah, I think there are some credits available for, related to this webinar presentation. Yeah, I would not know, I'm sorry. There's some training materials that are developed on GreenU, and some courses at Colorado State University, online courses that they've developed that have some credit related to managing plant genetic resources.
Okay, it looks like we are getting some questions coming in now. Here's one. What novel technologies are being used to assess the viability of seeds in the collections?
Yeah, so I wish our colleague Christina Walters, Dr. Christina Walters, was on that, because I know that they're trying to develop some technologies, novel technologies, to look at RNA degradation in seed lots without destructively sampling, the way we monitor classically germplasm collections for viability is germination assays. There's other techniques, but germination assays has been the go-to, and I know her research group is looking at being able to non-destructively sample seed viability and that decline in seed viability to catch it at a particular time for specific subgroups of related taxa. And so maybe that's the one answer that I can give right now.
Yeah, that's a good answer. Okay, here's another one is, do you have any suggestions or recommendations for people in developing countries to deal with climate change issues regarding germplasm preservation and conservation?
Wow, I don't think I'm at a place that I could answer that question. And, they could use. Go ahead, please.
Well, I'm thinking maybe there's two sides to that question. One is maybe there's part of it is a question of resources if you're in a situation with limited resources, and then a second one could be if you're in a location question. If you're in a location where climate change hits particularly hard, would you have different recommendations?
So one of our recommendations are, while introspecting on the NPGS, was to rely on climate, stations, weather stations, and to be specific for that site, to use that in decision making and to be able to maybe plug into our climate futures application. And so if those sites have access to weather station data, that would help them. If a particular site is being impacted, one of the recommendations would look at to see if there's alternative sites that do better for the collections that they're managing. But resource limitation is something that we all face, and that's a big problem to overcome in itself.
So here's a related question that I really like. do you have thoughts on assisted migration of in situ populations, maybe even as planned reserves in certain cases?
Wow, that's a ecological question. Again, I'm not sure that I'm the expert on assisted migration. I understand the concept, but I'm not sure that I have a good answer for that one.
It sounds like a cool idea.
What was the, can you help me out here? Is it that humans play a role in moving germplasm to more northern latitudes where it would do better instead of letting it on its own migrate over evolutionary time?
Right, I think that's the idea. And you could
in order to preserve it? Sorry.
You could create reserves that are more likely to survive and you know, give you another resource to tap into.
Yeah,
It's a cool idea.
I agree.
Okay, here's another one. Are you able to share info and understand protocols of the tribal nations in their efforts to save their native plants important for food and culture?
Would you mind rereading it or repeating it?
So the question is, have you shared information or protocol with tribal nations about what their efforts are to save their native plants and their food and culturally important species?
Yeah, we haven't shared this information specifically with any tribal nation, but I know that NPGS has efforts also somewhat based in Fort Collins, I think maybe in our headquarters office as well, on working with tribal nations to be able to pass on some of the technology transfer for managing plant genetic resources, yes.
I see a parallel there between, you know, the threat of climate change and the threat of, you know, cultural degradation. And it seems like maybe that parallel could be explored and maybe we could take some lessons, exchange lessons between the two situations. Okay, let me just run through and see if I've missed anything. Yeah, so there's a question about changing zones, and yeah, so this is kind of, I think, not just relevant to the germplasm collections, but it has to do with agriculture. So would changing zones where everything is planted help to avoid the climate change effects? Could some things be moved north or some things be moved south? Are we going to have to redo agriculture as a result of climate change?
That last question, or the part of the question, I'm nodding. I mean, it looks like we are going to have to rethink a lot of production agriculture, and what happens in production agriculture, there's corollary for what happens to the germplasm collections of those same crops. I know there's an effort, again, our national program leader, Peter Bredding, is involved in it to develop a new updated plant zone hardiness map, something that's cyclical, that happens every often with lots more information now. And so maybe those new and updated plant zone hardiness maps will shift things north. It's not easy to shift germplasm collections. You know, we have expertise, we have infrastructure, and part of the reason the gene banks at the locations there are now, that were chosen because the crops at that time were doing well there. And so in some situations, it might be easier to move things north. Think of Davis to Geneva, even though that's not a large latitude difference, the climate of the two locations is very different. And that's something that potentially could be done, but it's not very easy to implement across all the gene bank locations for many of the different collections. And there's only so far north that we can go with the crops.
Okay, and I think we have time for one more. It is about cryopreservation. Is cryopreservation necessary to maintain genetics and fight against unintentional genetic drift?
I think the last part of the question, cryopreservation doesn't necessarily help with genetic drift necessarily, and we can control that with other aspects of seed generated crops, like for example, those cages that were. But what cryopreservation does is preserve a particular genotype. And for clonal collections, oftentimes they either don't produce seed, garlic, for example, bananas, or if they do produce seed, it's going to be segregating. It won't be that unique genotype. Think of Cosmic Crisp apple. And so cryopreservation of these clonal materials are a way of preserving that unique genotype. And it's usually disease-free, and it's just a very effective way. Duplicating collections is another possible alternative to safeguard against loss. For example, there's a mango collection in 300-400 accessions in Miami, Florida, and a core subset are backed up in Puerto Rico. Unfortunately, it's very costly to maintain those collections in the field, and hurricanes, which we didn't touch on, could swing through Puerto Rico and affect Miami as well. And so cryopreservation is just a very effective, and it's not cheap. Initially, there's a lot of upfront costs, but once the material is cryopreserved, it's an effective way of backing up germplasm for the long term.
Okay, well, I think we're ready to wrap up. So I'd just like to thank you again, Brian, for joining us for today's webinar. And as a reminder, the recording will be made available to the attendees as soon as we can get it up. Liz posted in the chat the URL to the report that kind of stimulated this discussion, and you can find that there. I want to thank everybody for joining us again today, and have a good day, and goodbye, everybody.
Thank you.